Today I want to talk to you about stem cells, specifically liver regeneration. So who knows what stem cells are? Let's start with that. Oh, everybody knows stem cells.
I'll do it.
Go for it.
Yeah, so it's these cells that are very generalized, like they can grow in all different specific types of cells.
Right. So yes, excellent. That's the very baseline. We all know about stem cells. Stem cells are sort of the ground zero of cells. They can become any number of 220 different kinds of variety of stem cells. So what we're sort of working on is this idea of how to use stem cells, how to create them in the first place, and how we can best transplant them. So there are two different kinds of stem cells. The first one you mentioned, embryonic stem cells, right? Embryonic stem cells are collected from embryos. They are essentially the cells that will eventually grow into a person if allowed, and if not allowed, then they can be used for any number of other things. They are pluripotent, which comes from the Latin plurimus, which means very, very many, and potens meaning powerful. A couple people read that. So there are three primary germ layers. There's ectoderm, endoderm, and mesoderm, which basically define whether or not they're going to be gastrointestinal, neurons, muscular, bone, et cetera, et cetera, et cetera. So they can become any different kind of cell they need to. Embryonic stem cells are sort of where we started with stem cell research, right? And as we all know, there are issues with that. iPSCs are induced pluripotent cells, which means we basically created them. We were able to essentially take non-pluripotent cells, dermis cells, for example, skin cells, from a person, initially from a mouse, in fact, and then create them, basically re-engineer them into pluripotent cells. We were able to convince them that they were not in fact skin cells, they were in fact stem cells, which could then be turned into any other kind of cell that we want them to be. In 2006, a researcher named Yamanaka created the very first batch of iPSCs, which was huge. It revolutionized stem cell research. He won a Nobel Prize for it. Well, okay, let me ask you this. Why bother?
Republicans.
Right, okay. Accurate enough. Certainly there are huge issues with ESCs versus iPSCs, right? There are ethical issues, right? Some stem cells came from aborted fetuses. That was sort of that's the one that makes the press. There are lots of other ways of collecting them. But that's the one that, when there's nothing else to really piss off the radical right, we can always go back to stem cell research. There's always somebody who's trying to do something good using something that will offend somebody. So why not just always use iPSCs? The answer is, first of all, it's relatively new. We only really discovered the ability to make them in 2006. So we are still in the process of figuring out exactly how best to use them. They are very different from embryonic stem cells. The other issue is cost, right? Embryonic stem cells are available. They can be harvested, right? You can walk out into the street and pick yourself up a couple of embryonic stem cells. You can't do that with iPSCs. They have to be manufactured in a lab. It's incredibly expensive, and it takes a long time. So just out of curiosity, show of hands, is there anybody in the room who actually has an issue? That's a terrible way to present it. Does anybody in the room have an issue with stem cells? You? No. All right. So we all know sort of what the challenges are, right? We all know the conversation, the dialogue, and the iPSCs allow us to sort of sidestep it, but it's expensive. So this chart basically shows you the steps involved in taking a somatic cell, a mature cell like skin cells, for example, from an individual patient and then making them into iPSCs, then taking those cells to the next level and making them whatever else you need. So basically, so we have our somatic cells right here, right? This is a fully grown, already designed cell, right? This cell knows what it is. It's not a teenager. It has a life plan. It's going to be a skin cell, right? We reprogram that cell. We shake it up, a little, indoctrination. Low protein diet, take them out away from their families, make them wear white. Transduction editing, incredibly complicated, but basically what we're doing is we're reprogramming them. And then we wind up with these lovely, pure, genetically corrected iPSCs. which are sort of ground zero cells. They're now stem cells. We can now take what was originally a layer of your skin and turn it into a stem cell, which can now become anything else we need it to be. Differentiation, at which point we say to this wonderful ground zero blank stem cell, this iPSC, what we want you to become is, for example, a liver cell, or a brain cell, or a kidney cell, or a big toe. Not really. Quality control is really important, right? Any of you who have ever done anything where you run large series of numbers, large number anything, right? You're gonna catch 150 fish, you're gonna go out and shoot 150 basketball shots, you're gonna take 150 golf shots, whatever it is that you're gonna do, eventually you're gonna get some that work and some that don't work, right? Same thing applies to genetics. So if we start with 100,000 potential cells, each step of the process is going to reduce the number of viable cells, right? So it takes a long time, and the yield is fairly small to begin with. And that's one of the things that we're working on, is actually improving the yield. We want to make it better and more efficient, right? Now we've got a group of genetically matched healthy cells, and those cells know what they're supposed to be. In this case, we're working on liver cells. So now we have a bunch of cells that know that they're supposed to be liver cells, and they're from you. So we can transplant them into you if you need new liver. Okay, problem with iPSCs. The first problem with iPSCs are epigenetic markers, right? Once a cell, well, anybody know what an epigenetic marker is? No? Oh, wait, really? Yes.
Changes to genetics because of the life of the life.
Exactly. So epigenetic markers are emotional scars they had from being previous kinds of cells, right? It was a skin cell. Some part of it still believes that it's a skin cell. We're not actually sure exactly why that is. If there's 100% total knowledge, we have maybe 5% in terms of what cells do and why they do it. There's DNA, there's RNA, there's all these things that tell a cell internally what the cell's supposed to be, but there's a bunch of external information floating around the cell in terms of how it interacts with other cells, right? So yesterday, I was a skin cell floating along with other skin cells, and I was interacting with other skin cells as a proper skin cell should. Today, I'm a liver cell, except I still want to try to interact with other liver cells as a proper skin cell should. So that's one of our issues, right? So we're working on that one, but it's a challenge. Second issue is I can never say that word. Cancer. Cancer cells. Unfortunately, like many things, when you create something new or you take something that you have an existing identity and you change it, it doesn't always like that. Sometimes it becomes cancerous, it becomes tumorous. And the third issue is it's really hard to keep them, right? One of the most important parts of, oh, that's what it is. One of the most important parts, that's the fan. I was like, what is that noise? Sorry, I was drinking before. One of the most important parts of being able to have any biological treatment is scalability. We need to be able to mass produce it, right? It's great if it works one time in one small petri dish in one laboratory. Then somebody else in another laboratory a couple of years later is able to duplicate those results. Now it's science. Fantastic. It's still not necessarily viable as a treatment, right? So as you start to accumulate large groups of these cells, some of them will basically go bad. They change, they decay, they become cancerous. So it's not yet something that we can just stack lots of them together, keep them on a shelf, wait for somebody to come in who was in a horrible accident, give them a quick batch of stem cells and regrow their left leg. So why bother with iPSCs? This is a really wonderful word, autologous. iPSCs are created from the patient's somatic cells. So as I said, these cells are actually coming from you, right? If you need the liver transplant, if you need the treatment, these are coming from your body. Your body, the biggest challenge with any kind of transplant is that you're taking an outside thing and you're putting it into your body. Your body thinks that that is something that's not supposed to happen. It's attacking. So your body will basically try to reject the implant. It will reject the transplant, and your immune system tries to fight it off as if it were a disease. So in most cases, in all cases, they'll give you immune suppressants, which of course makes you vulnerable to any number of other diseases. So that's a challenge for general transplantation. And eventually the autologous cells will help us with that because they're your cells. Your body will recognize them and therefore it won't try to fight them off. ESCs or iPSCs from any other person's cells aren't immune privileged once they've been differentiated, right? So their stem cells, they're not going to be a problem. As long as they stay stem cells, you're fine. But the minute you tell that stem cell it's time to be a liver cell, it's time to be a brain cell, now they've been differentiated, now the body is going to react to them as a foreign body. It's also incredibly fucking expensive, like massively expensive. Hundreds and hundreds and hundreds of thousands of dollars to produce a very small amount of cells. However, this incredibly complicated little chart here talks about the next stage, right? So what do we do next? Now we have these stem cells. We've just spent several $100,000 to produce a small little stack of stem cells, iPSCs that we can use. The next step is to turn them into something, right? We have to program them. So we have to find out, well, what do we want them to be? A brain, lung, liver, stomach, pancreas, kidney, prostate. I didn't think anybody would laugh at that one, all right. So what we're doing is basically we're creating an organoid. We're creating essentially a framework. We're creating a stack. And it looks a lot like an organ, but it's not yet. However, the real value of these is that we can take these things and we can use them not just in the body, right? We don't necessarily just have to grow you a new liver and you a new kidney and you a new brain. Not that I'm saying anything about you. Right? But these organoids, this is really small, but these organoids can be basically anything, and they have phenomenal use not just in saving lives, but also in research, right? Unfortunately, you know, the Republicans won't let us crack a person's brain open so that we can study it properly while they're alive. So the best thing that we can do is we can create little... Don't tell me you don't want to do it, I know you do. Love that, magnets. All right. Brain organoids, right? We can model autism and spectrum disorders. We can model Zika virus infection. We can model Seckle syndrome, brain-specific region organoids, cortical spheroids. We can model cortical diseases. We can look at the way certain diseases attack your eyes, your liver, your spleen. And we don't have to actually go in and infect you with these things. We can actually just create them in a laboratory environment and study how they affect just that one area. Well, there's a long list here. I was going to read it all off to you, but eyes, lungs, kidneys, liver, cerebellum, cortical, gastric, intestinal, small intestinal. Incredibly, incredibly useful little things. They're sort of the Swiss Army knife of biology. So my research has been generally focused on, I'm working with a team, and we are specifically working on the liver. We're trying to create a viable liver transplant option. Right now it's still extremely difficult, extremely expensive. But liver disease affects millions of Americans. It's the 12th leading cause of death in the United States. Hepatitis, cirrhosis, liver cancer. You know, some of these are a little bit self-inflicted, but no sympathy at all. Shame on you. It's all right, I'm gonna fix it, don't worry about it. A lot of ways your liver can kill you. So, alcoholic hepatitis. It's an inflammation of or swelling of the liver, and I'm gonna read this off because I always get this wrong, and I've had a few. Basically, alcohol destroys your liver cells. It prevents your liver from functioning properly. 35% of heavy drinkers will develop alcoholic hepatitis, which can be mild or severe. Symptoms include fever, jaundice, that's when you sort of turn yellow, your eyeballs go a little yellow. Vomiting, abdominal pain, tenderness. Alcoholic liver disease is a term that encompasses liver manifestations of alcohol overconsumption. Cheers. Drink up, tip your bartenders. It's a major cause of liver disease, right? We have a culture where we drink socially, excessively. Most Western cultures, that's the major cause of liver disease. So, transplantation issues. We talked about this a little bit, right? Scalability. Number one problem is we need to grow a whole liver, right? The liver will regenerate, but if there's no healthy liver, right? Remember we looked at that, right here. If there's no healthy liver, I can cut half this liver off, but it's still a really unhealthy liver. It's not going to help. So if you get to the point where you actually require a liver transplant, you need a healthy liver. And you need a lot of it. So being able to scale those iPSCs up to a degree where we can actually use them to replace a human liver is what we're working on, but it's a real challenge. Also simplifying retrieval, right? So right now we can collect somatic cells from several different parts of the body, but it has to be retrieved, and it's also a timing issue, right? You can't scale them, you can't store them. So unless you know well in advance that you're going to need a liver treatment, but not so far in advance that you're going to need a liver treatment that you have time to grow them, but not enough time to let them die on you, it's an issue. Also maintaining homogeneity. The other issue is, of course, as I said, when you start to scale them up, when you start to stack them, you try to store them long enough to grow enough of them, some of them become corrupted, like data files, right? You have to clean sweep your computer every once in a while. They're just corrupted data files, and those files will corrupt other files, which will corrupt other files, which will corrupt other files, also known as cancer. However, fortunately for you, we can now make a replacement liver from your very own cells as long as you have lots and lots and lots of money. So that's the team that I'm working on right now. We are currently in the midst of a clinical trial to transplant liver cells from an autologous iPSC donor, essentially taking skin cells from you, making you a new liver. And right now, and this is the sort of really wonderful, exciting part of this, is that we have actually successfully transplanted liver cells that have fully vascularized and are functioning in vivo. Fully vascularized means that we have taken the stem cells that we've created from a person, we've put them up against their liver, essentially. We've connected them to a liver, and the vascularization means that the blood is now flowing from the original portion of the liver to the new portion of the liver, which is one of the hardest parts when you're basically doing a transplant. It's not as easy as just taking a half a guy's liver and another half another guy's liver, smashing them together and just hoping that they just go and just work, you know. It'd be awesome if it was, though, wouldn't it? It doesn't work that way. You actually need them to function. They need to recognize one another, they need to bond, they need to vascularize, they need the blood to flow back and forth between the cells, and then they need to function. So not only do they need to survive, the blood flow is what keeps it alive, but it also needs to function, it needs to do the job that we programmed it to do. That's what we're in the process of doing our clinical trials for, and so far, we've been very successful. So who knows, in a few years, if you have a couple of $100,000, you can drink all you want, and we can grow you a new liver. Thank you very much.